Alright guys, so welcome back. In this lecture we're going to talk about allosteric effectors. In the previous lecture we talked about the oxygen dissociation curve, and now in this lecture we're going to talk about the effectors, the factors that affect the shifting of this oxygen dissociation curve of hemoglobin. The hemoglobin curve will stay the same sigmoid shaped, but it can shift to the right or to the left depending upon the uh, change that the allosteric effector has brought. Right, so now let's get started. These are basically those factors which affect the affinity of the hemoglobin for oxygen. All right, they can change the oxygen affinity for hemoglobin, and they are called allosteric effectors because their binding at some site on the hemoglobin affects the binding of oxygen to the heme group on some other site of the hemoglobin. Okay, allosteric means other, right? So if these allosteric effectors, for example, if we talk about the hemoglobin structure, let me go back to the hemoglobin structure. For example, this is a hemoglobin structure. So in this hemoglobin structure, for example, we have the heme group right over here, and it is this point where the oxygen will come and bind, right? Oxygen comes and binds to the heme group. So what the allosteric effectors do, that they go and bind to some other site on this hemoglobin entire tetramer, some other position. They may bind to some other site on the tetramer, which will change the affinity of this site to accept oxygen. All right, so that's why they are called allosteric, because they bind to some other site other than the oxygen accepting site of the hemoglobin. Okay, now the most important ones are the partial pressure of oxygen itself. Okay, the oxygen concentration itself acts as an allosteric effector. The pH of the environment, okay, the proton concentration basically in the environment, which is actually dependent upon the partial pressure of CO2, the partial pressure of carbon dioxide in the tissues, and we are going to talk about this as well in great detail in the upcoming lecture. And the availability of 2,3 bisphosphoglycerate 2,3-ppg. This is actually an intermediate component of the glycolysis pathway, and this can also interact with the hemoglobin structure or hemoglobin molecule and changes affinity for accepting oxygen. Note that binding of the monomeric myoglobin. Okay, myoglobin is not affected by these allosteric factors at all. Okay, it will always have high affinity. This is a point to remember. Myoglobin will always have high affinity for oxygen. It will have affinity greater than the hemoglobin, but it will not be affected by, even if it is exposed to these allosteric effectors. Now let's talk about the allosteric effectors one by one in great detail. Okay, now the first factor is the oxygen partial pressure on hemoglobin. We've talked about this in somewhat detail in the previous lecture as well, when I was discussing the cooperative binding in one of those lectures. I'll give the link to that as well in the description. In that case, what happens is that the oxygen partial pressure on hemoglobin. So if oxygen partial pressure increases, the hemoglobin accepts oxygen. Okay, after accepting one oxygen at some certain heme group. For example, I have these four heme groups in a hemoglobin molecule. These four circles represent four different heme groups. For example, if I accept oxygen molecule has bound to this heme group over here. So what will happen? In cooperative binding, we learned that this heme group will change. The ferrous ion will move. It will I'll show you the diagram how it happens. We did this in the previous lectures as well. This ferrous ion, once it binds to the oxygen, it will move into the plane of the protoporphyrin 9, and once it moves into the plane of the protoporphyrin 9, so what happens, that this histidine, the F8 proximal histidine, which is attached to the ferrous ion through a covalent bond, it interacts with the remaining globin chain, and it causes the globin chain to move, and hence breaking this breaks some of the polar bonds. And if it breaks some of the polar bonds, then obviously the if it breaks some of the polar bonds, it interacts with the surrounding globin chain, and so attachment of oxygen at this heme portion, it changes the affinity of the adjacent heme portions of the adjacent hemes to accept oxygen. Okay, this is what we learned in cooperative binding, that these heme groups, they cooperate with one another in accepting oxygens. All right, so if oxygen has bound to this heme group over here, what it will do is that it will cause the hemoglobin, the globin chains to move, and this will break some of the bonds, the polar bonds in between them. And then once the polar bonds break, the dimers will start moving. And so if the dimer moves, so the globin chains, they lose up. Okay, they become loose. And once they become loose, the hemoglobin attains an R conformation. And so if it has to accept oxygen at the other site, at the other at the other uh, heme, at the adjacent heme side, it will accept oxygen more readily, because now in the R conformation, the affinity for accepting oxygen has increased. So again, this is an allosteric effect of oxygen. Binding of oxygen at this heme group, for example, this is the first heme group. Binding of oxygen at the first heme group affects the binding of oxygen to the adjacent and subsequent heme groups. So oxygen molecule got attached to some other site on the same hemoglobin structure, but it affected the property of the remaining three heme groups to accept oxygen. So this is an allosteric effect of oxygen to on the hemoglobin structure, and that's the reason why the hemoglobin curve appeared like a sigmoid, sigmoid in shape, and we've talked in great detail about that as well in the previous lecture. So binding, I'll just give a quick read through of what I've written over here. Binding of one oxygen molecule to the hemoglobin at one subunit causes the changes of to other subunits, okay, the adjacent subunits. All right, and this increases the affinity of other heme groups to bind to oxygen. Due to this cooperativity, due to cooperative binding, the affinity of hemoglobin for the last oxygen is as high as 300 times the affinity when it binds to the first oxygen. All right, so basically the last oxygen, we are talking about the hemoglobin when it has accepted three molecules of oxygen, meaning it has accepted six oxygen atoms, and it is accepting the last oxygen molecule to form to get fully saturated HbO8. So in while accepting this oxygen molecule, its affinity for accepting this oxygen is almost like 300 times greater than its affinity when the deoxygenated hemoglobin accepted the first oxygen molecule. So this is what it really means, and we've also discussed about this in the previous lectures as well. So oxygen itself is an allosteric effector that stabilizes the R form. Okay, binding of oxygen changes the hemoglobin structure into the R conformation, and it is stabilized due to the binding of oxygen. If this oxygen is removed, if the oxygens are removed, this R conformation becomes less stable, and then this hemoglobin starts changing its conformation into the T conformation, and we've talked about that as well in the previous lecture. Now, the cooperative binding allows more oxygen to be delivered to the tissues. Okay, it allows oxygen delivery at a faster rate to the tissues. For example, if this was the partial pressure in the lungs, and then in the tissues the partial pressure has become 40, what happens over here, small changes allow big drop in the percentage saturation of hemoglobin. We've talked about this in detail, but this is how it happens. Okay, and for small changes or decrease in partial pressure of oxygen, as shown by the steeper part of the graph. This steeper part shows that even if small changes, small decrease in partial pressure of oxygen is observed, the saturation of the hemoglobin decreases very steeply. In the lungs, small increase in partial pressure of oxygen allows more oxygen to be loaded by the hemoglobin. All right, so vice versa of what was happening first. Okay, so this is the effect of the allosteric effect of oxygen on uh, hemoglobin for binding to more oxygens. Now let's talk about the Bohr effect. Let's talk about the concentration, the effect of concentration of H positive ions on the affinity of hemoglobin to accept more oxygens. All right. So now what happens in the Bohr effect is that the Bohr effect, it tells us that whenever the partial pressure of carbon dioxide increases, okay, so what would happen is that carbon dioxide, when it increases the partial pressure, and this usually occurs in the tissues, whether the tissue respiration is very high, and as you know that carbon dioxide is a byproduct of respiration, so it is released. This carbon dioxide dissolves in water of the red blood cells. Okay, inside the red blood cells right over here, this carbon dioxide, it reacts with water. Okay, it's water. It binds to water inside the red blood cells, forming carbonic acid H2CO3. So it binds to water and forms H2CO3. This reaction is catalyzed by a zinc containing enzyme known as carbonic anhydrase. Okay, its cofactor is zinc anhydrase. Okay, and then this carbonic acid, once formed, it dissociates to form bicarbonate ions, HCO3, and hydrogen positive ions. Now, so if the partial pressure of carbon dioxide is high, this entire equilibrium shifts to the right hand side, and the H positive ions concentration in the solution increases. And if this increases, then the pH of the solution, it decreases inside the red blood cells. Okay, this is what we mean by increase in partial pressure of carbon dioxide in the tissue level. It causes the carbonic anhydrase, and then this carbonic anhydrase dissociates into bicarbonate and H positive ions. The bicarbonate ions which are formed, they diffuse out of the red blood cells into the blood plasma, and there they act as the major buffer of the blood. Okay, the major buffer of the blood plasma are the bicarbonate ions. They act as a major buffer. Buffer, as you guys already know, is a solution that resists changes to pH, and so this is responsible for maintaining a nearly constant pH of the blood plasma. I'm not going into the buffering properties of bicarbonate ions, but to discuss over here, the H positive ions which was produced, it does not diffuse out into the blood plasma, but rather this H positive ion binds to the hemoglobin. And now I'm going to discuss this part in great detail, but in a bit. Before going there, I'll tell you that this bicarbonate ion is the major form in which the carbon dioxide is transported from the tissues to the lungs. Okay, in the lungs, what happens is that the carbon dioxide, there is a gradient in that direction for the diffusion of carbon dioxide. So what happens, since bicarbonate ion concentration inside the red blood cells is decreasing due to diffusion of carbon dioxide outside of the red blood cells, the bicarbonate ions start diffusing into the red blood cells, and there the hemoglobin, it releases its H positive ions, and then this H positive ion combines with the bicarbonate, forming carbonic acid again, and then the entire cycle, it reverses, and then the carbon dioxide and water, they also dissociate, and then this carbon dioxide dissociate out into the alveolar airspace, from where it is expired out of the body. Okay, so it expires out of the body. Now, what effect does this H positive ions, which are produced due to the carbonic acid dissociation, have on the hemoglobin's affinity for oxygen? All right, so H positive ions can basically bind very strongly to hemoglobins. Okay, and they can bind to the deoxy part of the hemoglobin. They can basically bind to the deoxyhemoglobin more tightly than, and so if they bind to the deoxyhemoglobin more tightly, what they'll do is that they'll displace the oxygen molecules from the oxygenated hemoglobin structure. Let's look at this slide first. Okay, what happens is the carbon dioxide reacts with water, forming carbonic acid, and then which the carbonic acid spontaneously reduces H positive ions, forming bicarbonate ions. Now, the H positive ions which are produced by this pair of reaction, they contribute to lowering the pH. The differential pH gradient allows the oxygen to be allows the lungs to have a higher pH and tissues to have a lower pH, which favors the unloading of oxygen in the peripheral tissues and the loading of oxygen in the lungs. All right, so basically this change, the differential pH of lungs uh, being higher than the pH in the tissues, and I've told you that's due to the concentration of carbon dioxide and carbonic acid. This is the reason why oxygen is accepted mainly by the hemoglobin, and it is released by the hemoglobin over here in the tissue region. We'll discuss about this just a moment. First, let's go back to our initial slide of the Bohr effect. It tells us the decrease in pH increases the H positive ion concentration, okay, which decreases the affinity of hemoglobin for oxygen and allows the release of oxygen from the hemoglobin. All right, so H positive ions, they have a much higher affinity for hemoglobin, for basically the deoxyhemoglobin. So if oxygen is attached to hemoglobin, it must be removed so that the H positive ions can bind to the oxygen, to the hemoglobin, because of their higher affinity than the oxygen molecule. Increase in H positive ions occurs when the partial pressure of carbon dioxide increases in the surrounding tissues, and carbonic anhydrase dissociate. Carbonic acid is formed, which dissociates to produce more protons. We've talked about this as well. The deconformation of hemoglobin. All right, this is now a very important sentence that I'm about to uh, explain. The T conformation of hemoglobin is stabilized by the increased H positive ions, and hence most of the hemoglobin becomes deoxygenated. All right. So now, if the H positive ion concentration increases, it will combine with the hemoglobin, and if it combines with the hemoglobin, it will form HHB, hemoglobinic acid. All right. This is called hemoglobinic acid. What happens in this hemoglobinic acid is that in this hemoglobinic acid, the hemoglobin is in the T conformation. And whenever the hemoglobin is in the T conformation, its affinity to accept oxygen is very, very low. All right. So in the tissues where the H positive ion concentration is high, the hemoglobin, which is coming in the oxygenated blood, is rich in oxygen. It is loaded with oxygen, right? So what happens in the tissues is that I'll just take just a moment, guys. Okay, what happens in the tissue is that this oxygenated hemoglobin, it combines with the H positive ions, and it it releases its oxygen molecule. And once it releases its oxygen molecules, it forms the HHB. All right. This is because, as I told you earlier, that this H positive ion concentration, the H positive ions, after binding to the oxygenated hemoglobin, what they do is that they stabilize, or they cause the shape of the hemoglobin to change into the T conformation from the R conformation. Okay, and T conformation is the tense conformation, is the tight conformation of hemoglobin that has a lower affinity for oxygen. And since it has a lower affinity for oxygen, this causes this equilibrium to shift to the right hand side, and oxygen is released to the tissues, and tissues consume it. Now, what happens in the lungs? In the lungs, this entire equilibrium is reversed. In the lungs, the oxygen concentration in the surrounding tissue is high. What oxygen does is that oxygen then starts binding with the heme groups of hemoglobin, and once it starts binding to the heme groups of the hemoglobin shape starts attaining the R conformation back, and once it starts attaining the R conformation back, this H positive ions are released, and the equilibrium is shifted to the left hand side. Okay, H positive ion concentrations are released, H positive ions are released, and oxygenated hemoglobin is formed in the tissues surrounding the lungs, for the alveolar tissues around the lungs. Okay, so let's move forward. This is what this sentence meant. And then the increased release of oxygen by hemoglobin due to increased partial pressure of carbon dioxide and H positive ion concentration is the Bohr effect. All right. So Bohr effect tells us that if carbon dioxide concentration increases, the pH of the solution will decrease, and if the pH decreases, then the hemoglobin affinity for oxygen will decrease, and this in other words would mean that the hemoglobin would more readily release its oxygen content to the tissues, because partial pressure of carbon dioxide is high only in the tissues. Okay, it's not high in the lungs. In the lungs it's lower. So in the lungs it has to accept more oxygen, it does not release oxygen in the lungs. Okay. Now, conversely, the arc relaxed conformation of hemoglobin is stabilized by the high pH and low partial pressure of carbon dioxide. T form has low oxygen affinity and R has high oxygen affinity, and therefore the protons are called the allosteric effectors. Okay, they are also allosteric effectors because they bind to some other site on the hemoglobin chain, and they stabilize a certain form of conformation for hemoglobin, the T form, and this causes the oxygen to be lost from the heme site. If we look at its oxygen dissociation curve at a pH of 7.2 or 7.6, it's shifted to the left hand side. But if we look at the oxygen dissociation curve at a lower pH, the oxygen dissociation curve is shifted to the right hand side. Okay, So this is actually a right shifted curve, and right shifted curve would mean that the P50 of the curve has increased, and if the P50 increases, then what happens to the affinity? We've talked about this in the previous lecture as well. Affinity of hemoglobin decreases. Okay, so increase in carbon dioxide concentration increases the increases the H positive concentration. Increase in H positive concentration decreases the pH, and decrease in pH actually decreases the affinity of the hemoglobin for oxygen, due to which this curve shifts to the right hand side. Now let's give a read through for to this paragraph as well. The H positive ions which are produced by the pair of reactions contribute to lowering of the pH. The differential pH gradient, that is in the lungs, have a higher pH than the tissues have a lower pH. We know that pH and affinity, they go hand in hand. If the pH decreases, affinity decreases. pH increases, affinity for oxygen increases. So this would mean that the differential pH gradient, it will allow the unloading of oxygen in the peripheral tissues, okay, because in the peripheral tissues the pH is low, so affinity of hemoglobin automatically will decrease due to the entire mechanism that I've just discussed. And the loading of oxygen in the hemoglobin, basically the accepting of hemoglobin of oxygen by the hemoglobin in the lungs, will also be high because the pH over there is higher. If the pH is high, then the affinity for oxygen will be high. Thus, the oxygen affinity for hemoglobin molecule responds to small shifts in pH between the lungs and oxygen consuming tissues, and making the hemoglobin a more efficient transporter for oxygen. So this is how the H positive ion concentration affect the affinity of hemoglobin for oxygen. And now let's look at, okay, this the role of hemoglobin to accept H positive ion and to release H positive ion also makes the hemoglobin as a buffer inside the red blood cells as well. Because if the now the equation, the equilibrium reaction that I've just made over here, if this is actually a reaction of a buffer solution, if H positive ion concentration increases, then the hemoglobin will accept the H positive ion and will try to remove, and they will shift the equilibrium to the right hand side and will try to remove the H positive ions concentration from the solution, and they are trying to maintain the pH. And if the reverse happens, then the equilibrium shifts to the left hand side. So that's why it also acts as a pH. And this is the Bohr effect, is the mechanism of hemoglobin acting as a buffer. And H positive ions are accepted by specific histidine residues that become charged. Okay, if we go into more detail of hemoglobin, where do these H positive ions bind to the hemoglobin? It's basically to some histidine amino acid residues. Okay, histidine amino acid residues are present inside the hemoglobin. They accept the excess H positive ions and they become charged. And once they become charged, they form ionic bonds. And now, since you know that forming of ionic bonds would help stabilize the T conformation of the hemoglobin. So if the H positive ion concentration increases, this would mean that the pH is decreasing. And if the pH decreases, H positive ion concentration has increased, this will be accepted by the histidine. It will become charged. Okay, it will become charged, and once it accepts the protons, it becomes charged, and in between the charged amino acids, ionic bonds start forming. And these ionic bonds, they stabilize the T conformation. They stabilize the T conformation, and if they stabilize the T conformation, automatically the affinity will decrease. So an easier way of remembering the relationship of pH and oxygen affinity is that they go hand in hand. If the pH decreases, affinity will decrease. If the pH increases, affinity will also increase. Okay, so they will go hand in hand. So this is an easier way of remembering the effect of pH on the affinity for oxygen of the hemoglobin. Okay, so that's it for hemoglobin and the Bohr effect. And we've talked about the two allosteric effectors. Third allosteric effector is 2,3 bisphosphoglycerate, and we'll talk about that in the next lecture. Thank you so much.